In today's short lecture, we are going to cover respiratory pigments, as well as oxygen and carbon dioxide transport through the blood. We are going to focus in particular first on the respiratory pigments themselves, their characteristics, and then we shift over to explaining how these characteristics actually are important for transporting oxygen. We will then discuss the bore and root effects, which are important physiological phenomena that determine the amount of oxygen carried in the blood. Then we're going to talk about carbon dioxide transport, where we are going to learn that respiratory pigments are much less important really for the transport of carbon dioxide. And we will see that really it is an enzyme called carbonic anhydrase that drives most of the transport of carbon dioxide in the blood. So let's start with respiratory pigments and oxygen transport. Okay, hemoglobin. Everyone has heard of hemoglobin. This is the most common of all respiratory pigments, but it is not the only one in the animal world. Other animals utilize hemocyanins, chlorocruorins, or hemerythrins. Now, they all have similar functions to hemoglobin, but we're not going to talk about them here, but I want you to read up on those in your textbook. Hemoglobin is the most widespread respiratory pigment, and it has been around for a while. Now, some of you may recognize that 500 million years ago corresponds roughly to the Cambrian explosion when animals diversified. So right at the beginning of animal evolution, hemoglobin has been with us since then. Hemoglobin consists of two parts. One is a protein part that we refer to as the globin, and the other one is a heme group, which is a metalloporphyrin. Metalloporphyrin, let me just point it out right here. This is the heme group with the iron in its center. Here we see the heme group integrated into the globin. And finally, over here we can see that the hemoglobin molecule is actually a tetrameric molecule. Something very important, because it means that the transport of oxygen on the hemoglobin follows cooperativity. We're going to come back to that in just a moment. Finally, over here we see a graph that reflects a common experience, namely that oxygenated blood tends to look red and deoxygenated blood tends to look bluish, purplish. Now, other respiratory pigments have different colors. Hemocyanin, for example, cyanin meaning blue, is in a blue color. Alright, so a very important graph over here called the oxygen equilibrium curve or oxygen dissociation curve. What we see here is on the x-axis partial pressure of oxygen in blood, and on the y-axis we have the content of oxygen in the blood measured as milliliters of oxygen per 100 milliliters of blood. There are two lines on this graph. One, this black one down here, that shows us how much oxygen is actually dissolved in the fluid portion of the blood. So this is elemental oxygen dissolved in the fluid portion in the serum of our blood. But then we also see with this S-shaped curve here the total amount of oxygen carried by hemoglobin. Coming back to the partial pressure of oxygen in the blood. Now, how in real life do we measure that? Well, usually we look at mixed venous blood. We look at the content of oxygen in the blood that is returning to the heart in those very large veins just before they deliver the blood to the heart. If we measure the oxygen content right here, what we are doing is we're accepting the fact that the total amount of oxygen in the blood represents a sort of average oxygen coming back from the tissues. Some tissues gonna consume more oxygen than others. Obviously, muscles are gonna consume a lot of oxygen. Now, the mixed venous blood concentration depends on the rate of blood flow through the tissue, arterial oxygen partial pressure, the amount of hemoglobin per unit blood volume, and the tissue's rate of O2 consumption. All of those are going to determine how much oxygen is returning to the heart. Now, the oxygen dissociation curve is S-shaped, and this fact is actually very important. Why? Because we can see that over a large range of change in partial oxygen pressure, we see a relatively minor drop of oxygen. But as we are getting to the steeper part of this curve, we can see that a much smaller change in partial oxygen pressure is required to let go the same amount of oxygen from the hemoglobin. Down here in this somewhat steeper part, it's even less change in partial oxygen pressure to release 5 milliliters of oxygen per 100 milliliters blood. So we can see up here a large change in partial oxygen pressure is required to release 5 milliliters of oxygen from the hemoglobin, whereas towards the steep part of the curve, a much smaller change in partial oxygen pressure is required to lead to a similar 5 milliliter release of oxygen to the tissues. Now, a concept that I want to mention here is the blood oxygen utilization coefficient, the percentage of arterial O2 that is released to the systemic tissue. Now, here we have in fully oxygenated blood 20 milliliters of oxygen per 100 milliliters of blood. We release up to this point 5 milliliters oxygen per 100 milliliters oxygen of blood. This means that 5 out of 20 milliliters were actually released. So the utilization coefficient is 5 over 20. That's 0.25 or 25%. Down here at this point, the blood utilization coefficient would be of 50% since we got rid of 50% of the oxygen from the hemoglobin. Now, when we look at this curve in a slightly different context, asking ourselves, okay, what is the actual O2 concentration in the blood given the partial pressures of oxygen in different tissues? Now, obviously, in the lungs, we are going to have a high partial oxygen pressure. And that means that the oxygen concentration is also going to be very high. Now here, this is roughly the partial oxygen pressure in the blood as the blood moves through the lungs, or is coming out of the lungs. And at that partial oxygen pressure in the blood, we are nearly 100% saturated with oxygen. Once the blood moves out of the lungs back into the heart, and from the heart it is pumped to the systemic tissues. In the systemic tissues, the blood is going to encounter an environment that has much lower oxygen concentrations, and that is because our tissues are utilizing oxygen constantly. But when at rest, this is the amount of oxygen that comes out of the tissues that are at rest. We're talking roughly 40 millimeters mercury, roughly 5 kilopascals. At this particular oxygen pressure in the blood, we carry about 15 milliliters oxygen per 100 milliliters. So this is the amount of oxygen released to the tissues as our oxygenated blood moves through the tissues. But once we start to exercise, once we are utilizing our tissues, we're obviously going to utilize much more oxygen. And that means that the environment, the cellular environment in our tissues is going to carry much less oxygen. And at this point, we are releasing all this amount here of oxygen to our tissues. It's because, again, the amount of oxygen in our tissues that are active is going to be very low because our mitochondria constantly pull the oxygen towards them for oxidative phosphorylation. Once our blood moves through the tissues, when it comes back out, it's going to have a partial oxygen pressure that is going to be similar to the average oxygen partial pressure inside of the tissues. We lost this much oxygen. Now again, there is a fairly minor change in partial oxygen pressure from rest to exercise, which leads to a large release of oxygen to our tissues. And that is because of the S-shaped nature of the curve. Now, to give you some examples of how the oxygen dissociation curve may look like in different globins and different animals. Let's first look at this left-hand graph right here. Here we have in blue, our typical S-shaped curve for hemoglobin where cooperativity takes place. But we've also already seen another respiratory pigment such as myoglobin in our muscles. Myoglobin does not display any cooperativity, and therefore the shape of the curve is going to be very different. If we look at different animals, we're just comparing different animals, we can see there is a great diversity in terms of how the oxygen dissociation curves look like. In black we have the curve for humans, in red for the Weddell seal, which is an animal that dives, which means that this diving animal is able to carry up to 30 milliliters oxygen per 100 milliliters of blood, much more than we can. And that makes sense because when it dives, this particular animal cannot breathe, so it needs to take a good reserve of oxygen with it into the water. Then we can see a lot of poikilothermic animals down here with lower curves than the human one. And we see here reflected the general trend. Animals with a lower metabolism are going to have a lower ability to carry oxygen in their blood. It makes sense. Alright, so these are just a few things. We can point out a few others. Just think about the shape of the giant earthworm or the carp. Why is it different from, say, the bullfrog and the octopus? Think about the kind of environment that these animals are living in. Let's talk about the Bohr effect. It turns out that the oxygen dissociation curve is not constant. It actually changes a little bit. But before we get to the Bohr effect, I just wanted to point out two concepts. The first one, P50, and the second one, affinity, which are interrelated. The P50 is the partial oxygen pressure at 50% oxygenation. So we can look right here. Where are we 50% oxygenated? The corresponding partial oxygen pressure, that is the P50. Now the term affinity refers to the ability of oxygen to bind, the ability of hemoglobin to bind oxygen. That is the affinity. We're going to see that the two are actually inversely related. Why? Because when the affinity changes, the P50 changes. So let's look at this blue line. Let's say this is the amount of oxygen that hemoglobin is carrying at a particular condition. When this curve shifts to the right, we lower the affinity. A right shift of this curve means a lower affinity. Lower affinity means that our P50 has actually increased. So whenever the affinity becomes lower, the P50 rises. Whenever the affinity gets higher, the P50 drops. Now, in real life, when do we see such a shift of the partial oxygen dissociation curve? Well, we see it in our bodies whenever carbon dioxide concentrations increase in the blood, and we see it when the concentration of protons increases. In other words, when the blood becomes slightly more acidic. We can see that experimentally over here. The affinity for oxygen decreases in blood at 7.2 pH compared to blood that is at a pH of 7.6. So here you can see the more acidic the blood, the lower the affinity. There has been a shift to the right in the curve. A similar shift occurs when we simply increase the concentrations of carbon dioxide. So down here, this particular curve, we have a partial pressure of carbon dioxide of 10.7. This curve is much more to the right than these ones over here, particularly this one with a partial carbon dioxide pressure of 0.7 kilopascal. So increase the amount of protons, the concentration of protons, increase the concentration of carbon dioxide, and our blood reduces its affinity for oxygen. And this is very important. Why? Because it is in the capillaries that we utilize blood. It's also in the capillaries that through cellular metabolism we are increasing carbon dioxide concentrations. And along with carbon dioxide concentrations, we're actually also increasing the concentration of protons. So right where most of the oxygen is consumed, most of the CO2 and protons are produced. And this production of protons and CO2 actually helps us provide more oxygen to the tissues. That's what the right shift in the curve means. That's what a lower affinity means. We're providing or able to provide more oxygen to the tissues. Alright, so the next effect that I want to talk about is called the root effect, which is somewhat similar at first to the Bohr effect. We have a drop in pH which shifts these curves to the right. But not only is there a right shift in the curve, but we can also see that these curves become lower and lower, such that at a pH of 6.99 we can carry roughly 8 milliliters of oxygen on the hemoglobin. With a pH of 8.2, almost twice as much. So what happens to all of this oxygen that can no longer be carried on the hemoglobin? Well, it's released. It's freed up in the form of oxygen gas. So where do we need to produce oxygen gas? Well, it turns out in some fish, in the retina, in order to provide sufficient oxygen to the retina, and also in the swim bladder. The swim bladder that allows the fish to actually float inside of the water column. So how is this release of oxygen actually produced? Well, first of all, we have a particular organ called the gas gland. In the epithelial cell of the gas gland we produce lactate. Along with lactate we produce hydrogen ions, and all of these hydrogen ions are moved into the blood. when we move or increase the concentration of hydrogen ions in the blood, in this little equation down here, we're causing a shift to this side. We increase the concentration of this, and we shift the equation to the side, which means that now carbon dioxide is produced in the gas form. Carbon dioxide diffuses into red blood cells, where the same reaction takes place, but this time, because we are enriching the inside of the red blood cells with carbon dioxide, we're shifting the curve to this side, which means that now inside of the red blood cells we're producing more hydrogen ions which interact with hemoglobin. Hemoglobin now releases oxygen in gas form, which diffuses through the gland cells, the gas gland cells, into the swim bladder lumen. Pretty nifty trick to release large amounts of oxygen in an organ where we actually need oxygen gas. Now, in addition to pH and in addition to carbon dioxide, concentration, temperature, and 2,3-DPG are also important in determining affinity of hemoglobin for oxygen. Particularly, higher temperatures will reduce the affinity. Note the right shift in these lines, but note that unlike in the root effect, they all come back to roughly up here. So higher temperature, lower affinity, and some organic molecules such as 2,3-DPG, 2,3-bisphosphoglycerate, also affect the affinity of hemoglobin for oxygen. In this particular graph here we show concentration of 2,3-DPG, and on the y-axis we have the P50. We can see as the concentration of 2,3-DPG increases, the P50 also increases. And remember, an increase in P50 means a decrease in affinity. So it's a different way of representing this graph over here. Now in green we show the natural ranges for 2,3-DPG. Now I ask you, from where in biology do you remember 2,3-DPG? Think about it. And you will see that when we produce lots of 2,3-DPG, we actually do need more oxygen to our tissues. So in summary, Bohr effect, be it due to hydrogen ions or carbon dioxide, temperature, 2,3-DPG, they all enhance oxygen unloading, which means they all produce a reduced affinity for hemoglobin for oxygen. They all do. Let's move on to our second point, carbon dioxide transport, which is somewhat different from the transport of oxygen in the blood. Now, first thing that we need to remember, and here we see this equation again, which we saw previously on the slide explaining the root effect. Carbon dioxide dissolves in water, but it not only dissolves, it actually reacts chemically with water. Carbon dioxide and water produce bicarbonate and hydrogen ions. Hydrogen ions again. Now, how can we carry more carbon dioxide in the blood? Using this reaction right here. Well, we can carry more carbon dioxide in the blood in the form of bicarbonate if we somehow remove hydrogen ions from this equation. We remove hydrogen ions, and this equation will continue to shift over to this side, producing more bicarbonate. Now, it turns out that hemoglobin has a great buffering capacity due to the presence of histidines. And histidine, which is an amino acid, contains an imidazole side group. And it turns out that imidazole is a great base that will attract a lot of hydrogen ions and take them out of solution. Obviously, when you bind lots of positive charges to the imidazole, you're making them more positive, and introducing more positive charges on the histidine will start to cause three-dimensional changes in the hemoglobin. And it's those three-dimensional changes in the hemoglobin that explain the proton Bohr effect. Okay, so hemoglobin binds hydrogen ions, taking them out of the equation. The equation continues to shift to the right. It turns out that most of the carbon dioxide is actually being transported this way as bicarbonate. Some of it as dissolved carbon dioxide, and some to a much lesser degree by carbamate groups. Now, what are carbamate groups? Well, it turns out that some of the carbon dioxide reversibly binds to the hemoglobin by forming these carbamate groups. This would be the rest of the amino acid. This would be the carbamate group inside of the hemoglobin. Here we see a graph very similar to the oxygen dissociation curve. It shows us partial pressure of CO2 in the blood on the x-axis and carbon dioxide concentration inside of the blood. And we can see a very similar shaped curve. The higher the partial carbon dioxide pressure, the higher the concentration that we carry in the blood. There's also a fair amount of carbon dioxide transported simply as carbon dioxide. Now again, we can compare among different animal species, and it turns out again that in this particular graph, those animals that are the most active, that have the higher metabolism, also tend to have a higher capability to carry carbon dioxide in the blood. So again, this is related to metabolism, except for this turtle right here, but I talked about this turtle before. This turtle has the ability to persist for long periods of time in anoxic conditions. Remember? And obviously, anoxic conditions means that these turtles are going to accumulate a fair amount of carbon dioxide in their blood. Now, here with carbon dioxide, we have something similar to what is happening with the Bohr effect in regarding oxygen. It's called the Haldane effect. We can see that fully oxygenated blood is to the right of the curve for fully deoxygenated blood. That means fully oxygenated blood carries less carbon dioxide than fully deoxygenated blood. In particular, when we look at this figure over here, which just represents a subsection from this graph, we can see that there is a shift in the amount of carbon dioxide that can be carried by blood as we go from fully oxygenated to 70% oxygenated to fully deoxygenated. So again, as with the Bohr effect, as we're decreasing the amount of oxygen, we are increasing the ability of the blood to carry out carbon dioxide. So in many ways, the Haldane effect is the opposite of the Bohr effect. And right when we need to carry the most carbon dioxide, that's when we have lower oxygen concentrations in the tissues. It all kind of fits together. Finally, let's talk about this enzyme that actually catalyzes this reaction that we talked about earlier. Carbon dioxide and water make hydrogen ions and bicarbonate. So we can see that reaction taking place right here inside of a red blood cell. So we're producing bicarbonate and hydrogen. Hydrogen ions attached to the imidazole group of the histidine, causing hemoglobin to become overall more positive and provoking changes in the shape of the hemoglobin that affect its ability to carry oxygen, that affect its affinity, in other words. So with the hydrogen, we release more oxygen from the hemoglobin, which then goes to the tissues. Now, as we have less oxygen, we see the formation of a few carbamate groups. And as we drop the oxygen concentration, we are actually also increasing the ability of the blood to carry carbon dioxide. Okay, back to this equation. Hydrogen and carbon dioxide make hydrogen ions in bicarbonate. Hydrogen ions move to the imidazole groups. The bicarbonate is exported into the bloodstream in exchange for chloride. And this exchange of bicarbonate and chloride is referred to as the chloride shift. Alright, so now we're removing the bicarbonate into the bloodstream. In the bloodstream, we also have a few carbonic anhydrases in some tissues in the endothelial cells of the capillaries that will produce bicarbonate, even more bicarbonate, only that in the blood, in the serum, now the hydrogen ions are not buffered by the hemoglobin, but they are buffered by plasma proteins. So in this way, here we have the mechanisms by which we are actually producing bicarbonate using carbonic anhydrate, and we're producing more and more bicarbonate because at the same time we're removing the protons from the solution, and this process repeated again and again makes it possible for us to carry a lot of bicarbonate in the blood. And that bicarbonate is the way in which we transport the carbon dioxide. We reverse the reaction in the lungs. That is, we take hydrogen ions and bicarbonate and we get water and carbon dioxide, and in the lungs we're going to release that carbon dioxide. Alright, that's it folks for today. I'll see you later.